Here's the deal. Behold this. One of the alleged pieces of evidence that some people use to argue that COVID-19 doesn't exist. Basically, it's a document published by the CDC where they present their RT-PCR test to diagnose or detect coronavirus. Just to give you an example of two different posts saying the same thing about this. This person saying, more evidence that this made-up virus is from your own DNA, and this person saying, it's the most important video on Earth right now. Yeah, right. First of all, let me just start by saying that this is a horrible way to spread false information and lie to people who don't have a background in science. Because let's face it, this paragraph does look intimidating when you don't know anything about virology or molecular biology. Here, I've highlighted all the words that a non-scientist might not understand. And I can bet confidently that the people who are telling these lies don't even know what these words mean either. So, don't believe any random person just because what they say sounds sciencey. And that includes myself. Don't just believe me. If you have doubts, check my sources. Go through my logic. Correct me if I made a mistake. If a person makes a claim and just expects you to believe them without providing any valid proof, please disregard what they're saying and move on. Okay, let's take a closer look at this. This is the actual document, and I put the link in the description should you wish to read more. But if we look at the intended use section, they say the 2019 novel coronavirus RT-PCR diagnostic panel is a real-time RT-PCR test intended for the qualitative detection of nucleic acid in upper and lower respiratory specimens. What this means is that they created a test that detects viral nucleic acid, in this case the RNA of the virus that causes COVID-19, which is called SARS-CoV-2, and this test can be used to diagnose people. Now, there's a lot of debate going on about PCR tests, and I will make a detailed video about it, but in short, PCR is a molecular biology technique in which very specific sequences of nucleic acid, DNA or RNA, can be detected and amplified. Or in other words, many copies of a very specific piece of nucleic acid are produced, and this can be detected by a machine. So, in this case, the piece of nucleic acid that the test is assigned to detect is a region in the viral genome that, of course, we humans don't have. If amplification does occur, then you know the nucleic acid you were looking for was present, given a positive result, and if the machine doesn't detect anything, then you didn't have that piece of nucleic acid to begin with, and that would be a negative test result. Again, I will go deeper into this in the future because there are a lot of misconceptions about this technique. But for now, what we need to know is that PCR is a very sensitive test that can detect very specific DNA regions chosen by a scientist. And as any test ever invented by humanity is not perfect and it has its limitations. One of the ways in which scientists can determine how sensitive their tests are to make sure they're measuring what they should is called limits of detection. The limit of detection is defined as the lowest quantity of a substance that can be distinguished from a blank, or the absence of a substance, with a stated confidence level. Basically, how little viral RNA can you have in your test tube that your test or instrument can detect and that you can trust to be an actual positive. So now that we know that, let's see what's going on here. The first sentence says that this paragraph is about the limit of detection of the test the CDC designed. Now, this is what some people are getting wrong. It says here, since no quantified virus isolates of the coronavirus are currently available, assays designed for detection of the novel coronavirus RNA were tested with characterized stocks of in vitro transcribed full RNA of known titer. Honestly, I don't blame anyone for not understanding this the first time they read it. If we translate to regular English, this sentence actually says, because we couldn't use actual coronaviruses since they weren't available at the time, instead, we used the RNA of the virus to make this test as we did have many copies of it and knew the exact concentration of our stock. This makes total sense. Just think about it. You're a scientist in charge of developing an accurate test for a virus the world has never seen before in the middle of the actual pandemic. Virologists have ways of growing viruses either using animals or in cell culture dishes since viruses need host cells to survive and replicate. Then they can take these mixtures of cells and viruses and get rid of the cells, so all they're left with is a tube containing actual viral particles. There are ways to count these viruses so you know how many you have and use them for other tests, but all of this takes time to figure out, and these CDC people didn't really have that luxury. So, what's the next best way to make a diagnostic test? Using the actual RNA of the virus. We've known the SARS-CoV-2 genome since it was published for the first time in January 10th in China. It is now published on the NIH website, and no, it was not taken from human gene pool. The organism is SARS-CoV-2, and you can check this yourself using the BLAST tool. Anyway, using the viral RNA is a great idea to make a diagnostic test because both the viral particles and your cells will be full of this RNA if there is viral replication going on. So when you actually take a sample from your nose and do RNA extraction on it, you will get a combination of your own RNA, and if you're infected, the viral RNA, which will be detected by PCR. In addition to that, the CDC added a nice touch to the test design because they actually added some human cells and viral transport medium to the mix in order to mimic a clinical specimen. In other words, to see if their test could still detect the viral RNA even around all that human RNA. So, in conclusion, seriously, this paragraph does not say COVID-19 doesn't exist, and much less that the PCR test was made from your own DNA. If that were the case, there would be way more people testing positive, don't you think? It only talks about how sensitive their test is and what they did to create it. Don't believe me? See it for yourself. All the sources are in the description below. Please like and share this video so that it can reach more people. And to those individuals spreading this kind of information, seriously, if you have the time to go through this document, find the most random paragraph on page 40 and figure out an alternate meaning to it, you surely have time to look up what those words really mean. Just saying.